Hello everyone! Welcome to today's live broadcast, Standards for Microbiome and Metagenomics, supporting the commercial translation of microbiome science. I am Kristen Perret-Gentile of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. And if you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Jason Kralj. Dr. Jason Kralj has worked at NIST since 2006 and is a member of the Complex Microbial Systems group led by Scott Jackson. Currently, he is developing a pathogen DNA reference material to address some of the analytical challenges facing next-generation sequencing-based metagenomics. I will now turn it over to Dr. Kralj for his presentation.
Hi, as she said, my name is Jason Kralj. I'm from the Complex Microbial Systems group at NIST, and today I'm going to talk about our current and future work regarding the development of reference materials for metagenomic analyses for mixed microbial samples to support commercialization of microbiome science. My co-authors on this talk are Sam Forey and Scott Jackson. First, a big thank you to all the people who've helped us out over the last year plus, including our friends at FDA, Heike, Chelsea, and Brittany, as well as the other folks at NIST, including Nate Olson, and our friends out at LANL, MRI Global, Cosmos ID, and I'm sure there's many others I've forgotten, and I apologize. To get into it, who is NIST? The National Institute of Standards and Technology is a non-regulatory federal agency within the Department of Commerce. We've been around for a little over 100 years with main campuses in Gaithersburg, Boulder, Colorado, and we have about a staff of 5,000 scientists. Our mission is to broadly boost U.S. innovation and industrial competitiveness by advancing measurements, standards, and technology, which of course benefits the economy and everyone's quality of life. One of the ways we fulfill our mission is through the development of reference materials. Typically, these are adopted by community consensus. In this particular work, we interface with regulatory agencies such as the FDA and industrial partners to understand both sets of stakeholder needs to be able to address the challenges associated with NGS-based metagenomic tools and how to bring those to market. The first example where the FDA called upon NIST to support sequencing-based technologies was with regard to human genome sequencing. The FDA understood that if there was going to be certain NGS-based diagnostics, they needed to have confidence in the tool's ability to do exactly what they purport to do, and so they called upon NIST to help out with that effort. Dr. Zook and Salat helped lead NIST's effort in this arena and have had a major impact, especially in regards to accuracy of base calls within the genome. The combination of reference materials, the actual physical materials, and the reference data is what makes this such a powerful tool for the community. And of course, it never hurts when your work is highlighted in the New York Times, so NIST very much appreciates that. We also support the National Microbiome Initiative, or the NMI. That was just announced last year on May 13th, 2016. And it seeks to first answer fundamental questions about microbiomes in diverse ecosystems, to help generate insights to help share knowledge of microbiomes and enhance access to data, and to engage the community publicly through citizen science and education. There's been considerable attention and resources devoted to this space in the recent years, and our own group is certainly funded through part of this. In order to understand the community needs, we've engaged our stakeholders through workshops. Our recent one was our first one on microbiome-focused workshop was August of 2016, was attended by academic, government, and industrial stakeholders, and was a 2-day workshop with roughly 200 participants focusing on challenges all along the metagenomic analysis pipeline. As you see, I've got the link for the workshop up. If you want to go look, there's 2 days worth of recorded presentations. And about a year before that, in October of 2015, we convened a workshop to discuss pathogen detection issues. The report for this workshop is now online, and the summary from this is that by consensus we agreed that there was a clear need for reference materials. The strains we're going to select were going to be focused towards clinical applications, and that the particular aspects of which strains, were going to largely be driven by the challenges associated with the sequencing and identifying of those organisms. So looking a lot at near neighbors, high-low GC content, limits of detection and quantification, etc. The next workshop we have planned for this is going to be in August of 2017, and I'll talk about that more later. By that time, we'll have more materials to share with all of you. We're also working with the ATCC and FDA to ensure that we're striking the right balance of complexity while maintaining ease of use for everybody and satisfying the needs of the regulatory oversight. Meanwhile, we're going to continue to seek stakeholder input to ensure that we're staying true to the needs of this space. The FDA also recently published guidance on this topic, highlighting the fact that the components of NGS-based infectious disease diagnostics are subject to review. And as such, our interpretation of that was that NIST needs to play an important role in our reference material development. And that's mainly to improve the characterization of the analytical sensitivity and specificity of these systems, or as they're known as the FDA devices. Here we've outlined several factors that contribute to some of the 16S-based metagenomics uncertainty and even just general administration. Some of the parts, such as sequencing, can have protocols that are very nearly locked down and very well understood due to the kits and the automation that are currently available. At the same time, there are protocols and steps that can be quite unique to the individual tasks at hand. This is not just a necessity, this is oftentimes a very good thing. It's very targeted towards the application. And rather than tell people what they're doing right or wrong, we want to give the researchers the tools that they need to characterize each one of those steps to help them understand which parts of the process are critical to ensure reproducibility and to make their analyses portable to others. So these are some of the elements that help enable analytical sensitivity and specificity. And I should note this is by no means an exhaustive list, just many of the ones that sort of came to mind very quickly. And even looking at the whole metagenomics process more broadly, and this would include both shotgun metagenomics as well as 16S sequencing, there are multiple points where characterization and calibration are required. And clearly, our efforts are only going to address a portion of this challenge. So this condensed workflow identifies how we commonly translate a sample into something, into a report, or something we can understand or potentially even act upon. The organisms, looking at this workflow, the organism's DNA would be extracted, Purified, the nucleic acids would then be sequenced, and then the computational tools would attempt to identify which organisms each DNA fragment came from. And along this process, there are going to be biases or errors that are introduced and propagate throughout the entire analysis. In order to understand where and how these biases affect the end results, we work backwards. This enables the developers and evaluators to confidently focus as they go further and further upstream to what are known to be suspected or known regions of high variability within the process. Hence, we've chosen to start with DNA as the to analyze that for the sequencing and informatics steps. We realized that none of these mixtures, rather that any of these mixtures we create are likely to be artificial, simple, and unlikely to reflect what are real samples. Even so, this is a first step. And this process is designed to assess general performance. We're not trying to assess organism-specific identity at this point. Second is to acknowledge that this material is not going to help us characterize the pre-analytical variables such as extraction. And as you saw in the previous slide, there's a lot to be understood in this analysis moving forward. Eventually, NIST and our partners may seek to establish whole cell microbial mixtures that mimic and complement the DNA reference materials, which would likely be designed around understanding a lot of those pre-analytical variables. But we also know that the sequencing and informatics steps typically work well, although it's sometimes the limit of detection may be difficult to characterize, and there can be problems such as sequencing errors and weak database curation that will oftentimes plague certain analyses. Here I'm posting the list of our proposed reference material of bacterial strains. This list is subject to change. We're continuously evaluating the materials that are being produced to ensure that they're going to meet the stringent needs of our stakeholders and with both the purity and quality that everyone's expecting. Big thanks to the FDA, Drs. Campos at Children's Hospital and Chu at UCSF. They've been indispensable in helping us make selections for this list. And it represents a broad range of pathogens and commensals that can be signify or represent different diseases and taxa, including a lot of near neighbors, variable GC content, and even some drug resistance. And I want to emphasize here, this material was designed for pathogen detection. It's not intended as a mock community, and the deliverables for this are going to be vials of DNA, not whole cells. However, we anticipate the material can and will be used in a variety of microbiome studies because some or even many of the strains listed are likely to show up in a lot of the analyses and could serve as appropriate controls. To characterize the reference material, we're going to establish the DNA concentration, which will be combined with assembled genomes to help us estimate the genome copies per microliter or something to that effect. We'll do a base level purity and characterize the genomic contamination, and of course we'll characterize the stability. Much of this work will take some time, and there's potential for collaboration with sequencing centers and informaticians to help us parse these results and interpret them. We're making a modular panel of bacterial DNA in individual tubes. So when asked what is the mixed microbial DNA material going to look like, it's important to note that it's going to be left unmixed. We intentionally did that because that's what the community wanted, even though it's likely to introduce some heterogeneity and uncertainty in the analysis. The folks who are our stakeholders insisted that this was the best option for them. We're sure that people are going to come up with new ways to use this material, and this will hopefully allow researchers to extend the use of the material into areas that we haven't even thought of. So the idea here is that tubes 1 through 24 or 25 will be microbial strains, and then we'll also include a human genomic DNA component as part of the background. Looking forward, one of the future expansions of this is likely to be viral components. Most viral genomes aren't really an option to include here because of safety and export controls. So we're working with DNA manufacturers to figure out what can and can't be done so that we can distribute these without special licensing. It also highlights the flexibility of this modular design because we'll be able to incorporate new components to this while supporting the legacy components. And we also think that working closely with stakeholders of the NMI, other standards meeting more specific needs could also be developed, possibly with or including new materials, and incorporate best practices learned from the design of this set. To use the DNA reference material, you're likely to make some sort of mixture of DNA and work the samples up to run on your favorite sequencing or PCR platforms. You take that data and run it through whatever software you've designed to analyze it. And in this particular hypothetical, maybe you would mix a pathogen DNA with some human DNA, run it on a sequencer or PCR platform, or maybe both, and do some kind of response curve to generate an estimation of the abundance of the species or generate some kind of calibration curve for your protocol. The important part in all of this is the first one, that you have an understanding of the ground truth of your sample, of what's in it. It should become easier for you to interpret the results and identify the strengths and weaknesses of any particular analysis you would select. And there are several ways we envisioned the material could be used. Sequencing the isolates is one way to check the pipeline because it's simple and allows you to understand your process with specific organisms, but the real power is to be able to do mixture designs. So Latin square type experiments, which I've shown here on the right, where you can pool the organisms represented at different concentrations across different mixtures, which can provide not only a wealth of information about system performance, especially when it comes to limits of detection or limits of quantification. What we show in this sample experiment is a combination of equigenomic, is a combination of the equigenomic and log 10 dilutions, which one could multiplex on a single sequencing run even. And then you can start to ask questions like, can you detect organism X and Y? What about Y and X? Does the preference or presence of one organism even affect another's detection? What about spiking the material into a known material or even an unknown sample to help provide some sort of reference signal? In addition, these materials can be used to test the informatics as well, and these being the taxonomic ID methods and comparisons. Can we move beyond identity into quantification? Can we start to answer the kinds of questions that you see from regulators? So having well-characterized materials will allow developers and users alike to have confidence that the tools that they're developing have the analytical sensitivity and specificity that their applications require. And these are just some obvious examples. Certainly there are other complex mixtures that would stress the capabilities of NGS-based metagenomics analysis in a variety of ways. And we think the community is going to work hard to find many of them. When you dig into the data, as I'm sure many of you have, you start to realize there is a lot going on. We've tried to conceptually break this up into some of the components so that we can classify what's in the data. Some of it is going to be accurately sequenced, and here I've called this the real stuff, which is comprised of not only the actual sample that was in there, but also any contaminants and chimeric sequences that might have formed during library prep. On the other side, some of the data will have artifacts due to things such as sequencing errors or potentially mistakes or omissions in the database. And we know even some labs like to track trends with which instrument was used for sequencing, who prepped what, who did the run, etc., so that they can ID additional contaminants and false positives. And we think this is great. We think this is exactly what labs should be doing to sort of hone in on the uncertainty in their analyses. That's good measurement science, and we want to help support that. We would also help that the reference material will help all of you do the same and understand more, or maybe even potentially all, the sources of variabilities and bias in the informatics tools and sequencing. Clearly, our reference material isn't the last word, because it doesn't address a lot of the pre-analytical variables that we've discussed earlier, but it is a beginning. So here's an example of one implementation of the mixture design. We sequenced an equigenomic mixture of some of our existing reference materials, which contained 4 bacteria and 1 human genome, where we knew each of the individual abundances. We did these on an Illumina MiSeq using a Nextera XT kit. We took the data and ran them through 2 taxonomic identification tools. Here, using the BLAST plus BWA from Gotcha and Gotcha's species database to show how the different outputs can look when they run through different algorithms. And you see clear differences between the methods in that one gives more apparent false positives. Again, we know the 4 organisms that went into this, or rather the 5 organisms that went into it, but there's obviously more than that here. And not only the false positives, but have perhaps better sensitivity with the first one. So there seems to maybe be a trade-off, but both have false negatives. We know that we included Clostridium sporogenes in this, but it showed up in neither case. So you can very much get different results using different evaluation criteria. Looking at this data in a slightly different way, taking these same equigenomic mixtures, here I'm plotting relative abundance as best we could determine it using the output of each of the tool I considered. So here we have a list of 6, some of which I've redacted the names of. Normalizing the data to Salmonella enterica for only the organisms which we've selected. So we've excluded all of the false positives to just focus on sort of what we were looking at. The black bars represent what we input. And as you can see, there's general agreement between most of the tools of what was there, with the exception being Clostridium sporogenes, as we previously mentioned. When we followed up with the tool designers, we found that most of them did not include this organism in their database, so not surprisingly, it went undetected and was assigned and/or was assigned an identity of something else. And we think this kind of insight is a lot of what the community will encounter in their real samples. We can identify a lot of things we know pretty well, but there are still blind spots. And if a tool doesn't have a good match, you're either likely to get an unknown or a misassignment. And one of those is not necessarily better than the other. Having a tool that points to a particular organism could give a researcher a lead on where to look a little closer, maybe dig and find an organism that would have a better match, or lead you to other non-sequencing based diagnostics to follow up with. On the other hand, you may want a more conservative tool for applications where clinical misdiagnosis and mistreatment would have poor outcomes or bad effects. And all of this is to say that these simple mixtures don't always look so simple. The data aren't simple to interpret, and many of the microbiome samples that are out there are far, far more complex than these. And really, this is why standards help. Even though each platform may report different metrics, it's necessary that the sensitivity and specificity of the methods are well understood for objective validation of the performance. So NIST aims to deliver measurement assurance tools, standardized protocols, reference materials, validated measurements, and critically evaluated reference data. The rollout of this material will happen during the summer. We expect in June to take delivery of the purified DNA material, where we will do a baseline sequencing run on the material to ensure it is what we think it is, and start to assemble the genomes and perform some analysis of and characterize the concentration. We then plan to make the material available in August, though it won't be a reference material at the time. We'll continue to assess the material using deep sequencing. We'll certainly look to collaborate with people who get a hold of it, fully assemble the genome, and also to develop qPCR assays to further strengthen the quantification of the material going forward. Another major aspect of this material is that it works best when the entire community buys into it, so crowdsourcing. We want to work together through inter-lab studies for not only producing data, but to also help develop best practices in terms of mixture models and figuring out what suits the needs of maybe the majority or just a lot of the applications that are in this space. The reason being that reference materials are really at their most powerful when they're being measured many times in many ways by many people. And as such, we want to hear from you, in particular if you're a sequencing or bioinformatics lab, as we mentioned earlier. And as I also mentioned earlier, we have a workshop coming up in August. The purpose of this workshop is to present state-of-the-art pathogen detection technologies, emphasizing the needs for standards relevant to clinical, diagnostics, and biothreat detection stakeholder communities. Speakers will include subject matter experts in industry, academia, and government. And part of that will be to update on the progress of our DNA-based reference material. And part of that is we're hoping to send everybody home with a panel of our test material. So if you want more information, please follow the link. Registration should be available now, and shoot us a line if you need any help. And so to conclude, we know that there's a clear need for microbial reference materials for NGS-based metagenomics and detection. NIST will be making our first panel available in August. It will consist of 25 strains of bacteria plus 1 human, and is designed for pathogens, but will obviously include microbiome strains as part of this as well. The reference materials will help assess NGS performance of these microbial mixtures, targeting analytical sensitivity and specificity for, in this case, specifically pathogen detection, but obviously applies broader than that. Additionally, will enable evaluation of the existing pipelines for their suitability to handle these different kinds of samples. And finally, we want to be a community resource to all of you, and we want your interactions and your input. So please come to our workshop and contact us for additional collaborations. And with that, I would like to thank you and take any questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, how do you quantify low-level abundance using your materials?
So how do you quantify low-level abundance? Yeah, that's really one of the big questions that comes back to us. And the answer to that, I think, is largely it depends. There's a number of ways you could do it. You wouldn't necessarily need a reference material to do that. You could take a control strain that you have and quantify the DNA and dilute it down and run your sequencing analysis on that to try and assess where the limit of detection is for each specific organism. And I think that's important to note, is that each organism is likely to have its own particular limits of detection, especially with regard to not only the matrix that you're working in, but also the combination of other organisms that are likely to be present. So it's really a case-by-case basis.
Okay, and for our next question, what are your plans for including whole cells in the future? Will they be the same organisms?
So we know that there are a number of standards and materials out there already that you can go search on your own. We aren't looking to make those materials ourselves at this point. We are looking at working with some of the other companies who have their own strains to see if that would work. ATCC has expressed some interest, I believe, although don't quote me on that, in producing a reference material containing these organisms as well. I'm sure there are other companies out there who also have other mock communities that they would like to see incorporated in a lot of pipeline analysis.
If there are no more questions, I would like to once again thank Dr. Kralj for his presentation. Do you have any final comments?
None for me. Thank you for the opportunity to speak today, and good luck.
Thank you, Dr. Kralj. Today's webcast will be available for on-demand viewing through October of 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We'll see you next time. Goodbye.